Hey, good afternoon, everyone. Welcome to the inaugural CST 3-minute thesis competition. My name is Mike Zadilla. I'm a professor of chemistry here at Temple University, and it is my great pleasure to serve as your MC for Temple University's 2021 competition. So I'm very excited to participate in this event since we weren't able to do this last year due to the onset of the pandemic. But I'm very excited to be a part of it in this remote form and to learn about our 5 competitors' work. I also just want to say thank you to all of you who are taking part in the event. You've all gone through a long journey to essentially become the world expert on one particular topic. You've all made discoveries and uncovered a little bit about nature and the universe that no one else knew before, and on which you are the world's expert. And that's really something quite incredible and something to be very proud of, regardless of the outcome of the competition. So just a few words about the 3-minute thesis competition. This tradition began at the University of Queensland in Australia. The original idea came about during a drought, during which citizens of Queensland were encouraged as part of a larger sense of responsible citizenship to limit their showers to 3 minutes to save water. And so everyone had these egg timers on hand to time their showers. And in a spirit of solidarity, the dean of the graduate school, Dr. Alan Lawson, had this idea to translate this 3-minute aspect of life to a thesis competition where folks would describe their thesis idea in the same amount of time that they had to take their shower. So the idea has really taken off, first in Australia and then New Zealand, but then it spread and eventually became an international phenomenon that happens at institutions and conferences all over the world. And it's my great pleasure to present to you the inaugural Temple University CST competition. But before we get started, there's a little bit of business to take care of. There are some rules that we want to go over. So the presentations are limited to 3 minutes, exactly 3 minutes, and there will be a timer. When it gets to 0, each competitor must stop speaking. There will be one PowerPoint slide per presenter, so there's no animations, no transitions, so everything that each presenter will talk about will be on one slide. There's no additional augmentation by electronic media, no sound or videos, no props, costumes, instruments, sound effects, anything like that, or laboratory equipment demonstrations of any kind. And the presenters are to use spoken word only, no poems, raps, or anything inspired by the musical Hamilton. Next, I want to go on and acknowledge and thank our judges. So our judges are Dr. Sherry Gillespie of the CST Board of Visitors. We have Dr. Amy Defabau, who is the director of CLA Graduate Affairs, and my colleague, Dr. Serge Jasmine of the Department of Chemistry. So I want to thank all of you for taking the time to support these competitors and to serve as our judges. And I want to invite everyone in the audience to play a role as a judge in the Viewer's Choice Award. So at the end, you will all get a chance to vote for your favorite competitor as well, who will be acknowledged by an additional award. Okay. So with that, we are ready to begin our competition. And our first speaker will be Louise Borthwick. And the title of Louise's thesis talk is What Lies Beneath? Using Geophysics on Thwaites Glacier. And so go ahead, Louise, take it away.
This image is taken on Thwaites Glacier in Antarctica. Antarctica is this huge expanse of ice sitting down at the south of our planet, and Thwaites Glacier is one of the most unstable parts of it. The complete melting of Thwaites would raise global sea levels by 3 meters. That's enough to cause Old City, the airport, and the Schuylkill River Trail to all be underwater. What we don't know is how fast this ice is going to melt, or more exactly, how quickly it will flow into the ocean. And one of the biggest problems is that what we see here on the surface of the ice isn't going to tell us much. It's what lies beneath that's really important. Both the topography and the types of rocks that are down there are going to have an impact. And what's going to happen is that on bumpy mountainous parts, the glacier will melt more slowly, And on flat, smooth basins, it will melt more quickly. So how do we find out what's down there? Do we dig it up? Well, Thwaites Glacier has ice that's 1.5 kilometers thick, and it's around the size of Florida in extent. It's huge. So what I do in my research is I use geophysics to try and understand what's going on underneath the ice over the whole glacier. And specifically, I use two techniques, gravity and seismics. So you might not know, but gravity actually changes over the surface of the Earth. And it changes depending if we're near the equator or near the poles, depending if we're up a mountain or in a valley, and crucially for my research, depending on the density of the rocks in the Earth's crust. So we can fly a plane over the surface of Thwaites and see how the gravity changes. And that's going to help us find those flat, smooth basins which allow the glacier to melt very quickly. But the problem with gravity is there isn't just one thing that causes a gravity value. What we think is a smooth basin could actually be a change in rock type, which therefore shows a change in density. So we're bringing that second technique, seismics. And with seismics, we set off an explosion at the surface of the ice, and the waves that originate from that explosion penetrate all the way down to the bottom of the ice, maybe into the basin if it's there. They reflect off the bottom and they come back to the surface where we record them. And this gives us improved confidence that the basin is actually there. But the problem with this technique is we can't do seismics from a plane. We can only collect this data over a line of maybe 40 kilometers, not much when you're trying to understand a glacier the size of Florida. So I combine these two data sets and I link them to larger scale patterns in the topography underneath Thwaites. So far, I've found smooth basins in at least 2 locations with more mountainous areas between them. These improved constraints on topography and geology will help us understand how much Thwaites will contribute to global sea level rise in the next 100 years, and therefore how quickly we can expect this ice to have an impact on our lives.
Thank you, Louise. That's really interesting, and what a striking image. So this concept of measuring gravity while flying over in a plane is a new concept to me, and that's really fascinating. How does that work? Is that just an accelerometer of some kind? And how do you take out like small motions in the plane from those measurements?
Yeah, so it's a special instrument called a gravimeter. And yeah, it works, the basis of it is like a mass on a spring, and you see how much the mass gets like pulled up and down on the spring. And yeah, there's a lot of corrections to be done, especially when you take measurements from a plane, because you're right, like the plane moving up and down changes it. So you need to try and have as steady a flight path as possible. And then there's still a whole load of corrections you need to make to try and find these tiny, tiny changes that we're looking for that come from the rocks.
That's really fascinating. I would have never guessed you can tell what kind of rock is below you by sensing the gravity. That's really fascinating. The other thing I was wondering about is, so when you use your seismic approaches, you're basically trying to correct for changes in density versus changes in total volume of rock. And are you guys able to know what type of rock, like what material is down there based on the density, or do you just know this is a more dense place and this is a less dense place?
Yeah, so all the gravity tells you is more or less dense. So that's why we try and bring in the seismic, and you can get kind of an extra constraint, but it's still very hard to tell exactly what rock it is. It's really like the properties of the rock that are interesting to us. So we basically need to know if the ice is going to move faster or slower over it.
Is the density of the rock going to figure into so if you do the modeling for melting rates, does that end up being important, or is it just much more the planarity of the surface?
It is more to do with what the density of the rock tells us about. So less dense rocks are more likely to be sediments, which are therefore more smooth than like more basement rocks, which would be more the mountainous areas.
Wonderful. Thank you so much. That was great. Thanks so much for the presentation. So at this point, we'll move on to our next presenter. So our next presenter is Jay Padil from physics, and the title of Jay's talk is Magnetism from Non-Magnetic Materials Interface for Future Generation Electronic Devices. Go ahead, Jay.
Okay, so finding a new material for the technological development is always important. If we go back to the 80 years ago in the past, So we can see that at the beginning of the birth of the computer were of the size of a room. So one single computer occupied the single room. But nowadays you can see that the computer can be fitted in our palm and hand. So isn't that amazing? Yes, definitely. So this is possible because of the development of new materials for the magnetic storage and also the transistors. So the race of technological development is not over yet. So we still try to improve the performance of electronic devices in terms of their speed and also in terms of their memory storage capacity. And also we want to decrease the size even smaller depending on our needs. So how that can be possible? So for that, we need to investigate and do the research for the new materials. So from 20 years ago, starting from 2000, people are studying this kind of interface phenomena. You can see it in the top middle panel. So that kind of interface phenomena is hugely studied. And my project is related to the two different kind of oxide sandwiched together. And then at the interface, there will be the magnetism appear from the non-magnetic parent material. So that will be very, very interesting, that magnetism obtaining from the non-magnetic materials. So for this study, we use the X-ray spectroscopic technique. Let's compare this with when we go to the hospital, doctor orders the X-ray and investigates the X-ray image and confirms what's going on inside our body. Similar to that, we do the X-ray of the samples in our lab and then get the image and process the data and found the results and then confirms what's going on inside there. So on the rightmost panel, you can see some results that the peaks indicates that there is magnetism at the interface. So that the obtaining peaks indicates that there is magnetism. And we also already quantified how much thickness is magnetized. That is 3.34 angstrom. So let's let's compare with the real object. So if we compare this with our human hair, it is 10,000 times smaller than the dimension of our human hair. So isn't that cool that this much thinner materials can be applied for the magnetic memory devices? So my future project is to control this magnetism for the future generation electronic devices. Thank you.
Thanks, Jay. So that's really amazing. So it seems like what's happening here is you got two things that aren't magnets, but where they touch, you get a magnet. Is that right?
Yes, that's very interesting. We obtain the magnetism from non-magnetic materials when we sandwich them together.
Right. And so this X-ray imaging technique, is that actually an absorption technique just like you see with the hands, or is this actually a diffraction or a spectroscopy?
Oh yeah, that's the image that I showed here is the photo emission technique. That means when the X-ray hits on the sample, electron come out from the sample, and we record the electron in the analyzer that you see on the middle panel, middle figure, the shiny things. That is the analyzer, and so that detects the electrons, and we got the image. However, as you already mentioned, that absorption technique that I didn't show here, but we used to travel national lab in Switzerland and also in Berkeley and collect that kind of data as well. But this is photo emission.
Right, I see. So this, you're measuring a photoelectron or
yes.
an X-ray fluorescence?
Yes, photoelectron. This is photoelectron, but we also measure the absorption data in the different lab.
Right. So the other thing that I noticed that really struck me is that if you look at the right panel with the pink and yellow plots,
yes,
there's a difference between where the ruthenium is on the left and the manganese on the right, and then the next junction with the manganese on the left and the ruthenium on the right, they're not the same. So it seems like the magnetism is not the same on the two sides. Is that true? And why would that be?
Yes, exactly. That is the new thing that we got now, and we are collaborating with the theorists to find the explanation of this. And that might be because of polarization catastrophe. That is our guess, but we haven't confirmed it. We just quantified the magnetic thickness, and we want to investigate that asymmetric magnetism at the interface. And that might be because of polar catastrophe, because there is a substrate. And but we have to confirm that, and we are collaborating with the theorist to find the explanation.
Right. Okay. So it sounds like you're suggesting that at least one of these oxides has to be a polar crystal structure.
Yes.
A thermoelectric or something.
Yes.
Wonderful. Thank you so much, Jay. That is a really surprising, as someone who studies magnetism, a pretty mind-blowing result. Thank you for sharing that. At this point, we're going to move on to our next presentation from Avik Bishwas, which is evolution of drug resistance in HIV. Avik, go ahead.
Am I audible clearly?
Yes, sound good.
All right. So last year, in the middle of an ongoing pandemic, the World Health Organization declared HIV/AIDS to be one of the biggest pandemics in the history of mankind. So today, although we have antiretroviral drugs to treat HIV/AIDS, it is estimated that roughly 50% of the people who are taking these drugs today are going to develop resistance as early as 2025. Now, this makes tackling the drug resistance problem in HIV one of the major challenges. And my thesis is focused on understanding how this resistance develops in HIV. So to do that, we start by first collecting thousands of HIV viral sequences from patients who are grouped into two categories: one who have taken drugs and one who have not taken the drugs. And then we build sophisticated machine learning models to understand and identify the patterns of similarities and dissimilarities between these two groups, which can help us in understanding how this resistance arises. So how does resistance arise in HIV? HIV has resistance primarily caused by certain kinds of mutations, which are called primary mutations. Now, when these mutations, however, first occur in the virus, they're not well tolerated. This is because they carry a penalty to the fitness of the virus. So for example, a virus with such a mutation may be able to reproduce or replicate at a much slower rate than the original virus. However, with drug pressure, changes start occurring in the rest of the viral genome and the rest of the viral sequence, all of which act in concert to really stabilize and accommodate this primary mutation. And this may go on up to a point where it becomes difficult for the virus to revert the primary mutation back to what originally was there. We then say that this mutation has become entrenched in the virus. Now, this can have serious consequences. One consequence is that a patient who is carrying a virus with such an entrenched mutation is likely to transmit preemptive resistance to someone who has not even begun drug therapy. And the second consequence is that studies have shown that certain drugs select for particular patterns of resistance mutations. Now, what happens if these mutations are already entrenched in the patient? So the future goal of this study is to identify and understand if such patterns of primary resistance mutations already exist and are entrenched in drug-naive individuals. And if any of the new drugs that are coming into the market today will select for these mutations and these patterns, in which case resistance will arise much faster than otherwise predicted. Thank you.
Thank you so much. The first thing that struck me, and maybe this is a little bit of a depressing thing to bring up, how hard is it to find subjects who have HIV but have not been taking drugs?
Well, this is the interesting point. With HIV, since there wasn't drugs available for a long period of time, you actually find thousands of sequences which are actually stored in public databases, for example, from Los Alamos and Stanford, which are pretty well curated, and you can get those sequences. Also, the key thing that we have to know is that for HIV, whenever a person gets tested positive for HIV, he or she undergoes what's called a drug resistance testing. And they check for mutations that may sometimes be present in the patient. And then according to that, a cocktail of drugs is prescribed. Now, before this drug prescription. So the person has already gotten tested. So you also have these person sequences who has not yet started taking the drug.
Thank you. Thank you. So these drugs, so you put drug A, which seems to suggest you're looking at a couple different drugs in this study. Do these drugs all work in a similar way? Like, do they all say attack the reverse transcriptase, or are these drugs different in their operation?
Yeah, that's a good question. So no, these drugs are not all the same. So it's different drugs attack different components of the HIV life cycle. So some attacks may be reverse transcriptase, some attacks the integrase protein, which binds the viral DNA once it's been reverse transcribed from the RNA to the human genome, and also attacks the protease. Now, the point, however, is that we have now more or less come to have a stalemate as to find newer targets in HIV. So this makes the drug resistance to all these existing targets a major problem, because we are already seeing resistance to these drugs. Unless we do something about resistance or find new targets, that's going to be a major problem.
I see. So this type of knowledge can help you to identify new targets?
Yes, new targets as well as modify pre-existing drugs in certain ways that they may not select patterns which are already present, or something that will give rise to resistance faster. So buys us some time.
I see. So they could attack the same targets, but in a slightly different way.
Right.
Thank you so much, Avik. Very nice work.
Thank you.
Let's go ahead and move on to our next presentation from Rajpreet Gulati. And the title of Rajpreet's presentation is Magnetic Induction Communication in Challenging Environments. Go ahead, Rajpreet.
Hi everyone. So my topic is magnetic induction communication, communication in challenging environment. So it's basically called, which everyone knows about, magnetic induction. So it is just like a mutual induction. So which is a basic principle. So here it is power is proportional to I square, and the induced powers decay by as r raised to power 6, and it has a source. It's a secure and no leakage, and scattering absorption is very small. So that's why we are going to use this magnetic communication in a various media, and air and air, human body, and and live tissues. So we are going to do that kind of experiment. So basically why we need that, and why we are going to use in the human body, because this has a no leakage, and also as we know that in US 45% or 133 million of all Americans suffer from at least one chronic disease. So we want to monitor everything, like from the from the human body, to check that pH and oxygen and respiratory and that kind of things. So and also we want these sensors what we are making up it in for the magnetic communication or magnetic sensors. We want that it should has a can be used in one of the application, we can say in the broad-minded way, in endoscopy, where we can say that TX is in our hand and RX we can keep it up in the mouth to get the whole picture of the body. So we can be used in that. We can use this sensor in the on body to check the pH and oxygen values as well. So yeah, and as I have done various experiments with the air, tap water, and aluminum and magnet and human as well. So where that the blue one is showing that my sensors are working up to a 3 meter, 3 centimeter, 3 meter up to like 3 meter in air. And similarly, I have done a various different media like human and chicken. And as you have seen in the second figure, that chicken test is like when we have done chicken test, put the sensors in the TX and RX, and the
Okay.
power is more. Yeah, so
Okay,
that's it.
great. I'm going to have to stop you at that point. Thank you so much for sharing these results. So it sounds like the idea here is to figure out how signals will travel through these different media for some sort of in sensors that would be implanted like medically in a patient. Is that right?
Yeah.
Right. And so what is the actual signal? Is it radio waves that we're using to excite the magnetic induction?
Yeah. It's like any you can say that they are type of radio waves.
Okay. And so I guess what was surprising to me is am I reading the plot correctly in the upper right that a sharper slope means poorer range, right? So it seems like it travels better in a chicken than it does in the air. Is that right?
Yeah, that is so that's why we are going to use in the implantable, because we are thinking that when it is these sensors are going to be implanted in the body, because this is a low frequency sensors as compared to the radio frequency, because this is working on a 13 megahertz frequency. So we have used that again in the chicken and in the implantable body, or we have used this sensor in my body as well nowadays. So and yeah, and we measure that with the human tissues. These sensors are working pretty much as compared to air, because air has a clutteration and that kind of behavior.
Are there other wavelengths that work better in air? I thought radio waves were meant for the air, so that's kind of an interesting and surprising result.
No, this is not a particularly I cannot. This is a low frequency. This is not a radio waves. This is 13 megahertz.
So this is a different wavelength.
Yeah, different wavelength. This is 13 megahertz. It's working. The radio waves is more than gigahertz.
Right. Okay.
So this is not a radio waves. Okay.
Got it. Thank you so much, Rajpreet. Thank you for the presentation.
Mm-hmm.
And now we're going to move on to Melissa Betters from Biology Department, who is going to present the talk entitled Investigating Deep Sea Snail Species at Chemosynthetic Environments. Go ahead, Melissa.
All right. My first dive to the bottom of the ocean was in 2017. I was on a research cruise in the middle of the Pacific Ocean, miles away from the nearest land. That morning, the sub-pilot, myself, and another researcher climbed into the submarine and descended to the ocean floor. And it was like landing on another planet. This environment was completely foreign to me, but it wasn't for the sub-pilot. He was able to tell us what almost everything we saw was. And what's more, he was able to identify if something was new or interesting about what we saw. Like he'd never seen that fish this deep before, or he didn't know that coral lived on this seamount. It became clear to me that his ability to recognize these species is what gave him the foundational knowledge to make these observations. The process of classifying organisms is a branch of biology known as taxonomy. And it's what I do. I work in a very particular environment in the deep ocean, known as chemosynthetic environments. At these environments, tiny organisms are able to create food and energy directly from chemicals released from the ocean floor, meaning these are some of the only organisms on Earth that don't rely on sunlight for their food. These organisms coat the hard surfaces at these sites, and the animals I now study, the snails, eat them. My work looks to identify the huge diversity of snails at these sites. The problem is these environments are really hard to get to. So how do I find new species to classify? Well, we could just scour the ocean floor and hope to find something new, but that would obviously take a huge amount of time and effort. A better way is to predict where new species are most likely to arise and to look there. For instance, on land, organisms on either side of a river or a mountain range will most likely be different species, particularly if those environments or if those organisms can't cross those barriers. Identifying barriers like this in the ocean, though, can be pretty difficult. So my research looks to find what barriers do exist. Is water depth a barrier that animals can't cross? How about water temperature or the concentration of dissolved chemicals? I look at snails on either side of these potential barriers, and by comparing their DNA, I can tell whether these barriers may be driving these organisms apart or have sometime in the past. This provides myself and others with a blueprint on where to look for new species in the ocean. Documenting the vast diversity of life in our oceans is daunting, but the more we can tell about marine species, the better equipped we'll be to protect and manage them for years to come. Thank you.
Wonderful, Melissa. Thank you for the presentation. I was really struck by your comments. You seem to be quite amazed by the subdriver. What is the subdriver's background? Is this person a scientist?
Yeah, so this person was actually not technically a scientist, but I mean, of course, in my mind, he is. You know, he had been piloting the submarine since the mid-1960s. And his ability to just recognize these species, you know, any one of the observations that he was able to make could be an entire research project. And that really struck me, which is what got me interested in, you know, the field of taxonomy and classifying organisms for the use of other researchers.
That's really interesting, and it speaks to my experience that you find brilliant people in unlikely places. You know, that
Absolutely.
We tend to think as scientists, as PhDs, that we're the smartest folks around, but once in a while you just meet someone and you're like, how are you not, you know, how are you not an MIT professor or something like that?
Yeah, sometimes that practical knowledge really goes a long way.
Yes, for sure. How deep are you going on these subdives?
Yeah, so the deepest that I went was about 1,600 meters, which is, I'm not sure what that is in feet, around between half a mile and a mile down, I think. But these chemosynthetic sites that I study, I have specimens as deep as 3,000 meters that I'm looking at.
It's really, really amazing. Must be scary the first time you go down.
Yes. Quite an experience.
So as I think about this concept of undersea barriers, or I guess some of them are oversea barriers, or maybe all of them, but so it sounds like these snails have to be like near undersea vents where there's a lot of minerals being expelled. Are long distances between like tectonically active areas, are those barriers, like can these things cross from one vent to another, or is that considered a barrier?
Yeah, so that's exactly what I'm researching. We don't necessarily know. We do know that they have, you know, mobile larvae. So they do have a larval stage where they can travel quite a distance. What that distance is, we are not sure. But we do know that these chemosynthetic sites can occur, you know, hundreds of kilometers apart from each other. And we still find, you know, these opportunistic snails there. So figuring out, you know, how species arise in a seemingly barrier-less environment is exactly what I'm trying to figure out.
Are there other less physically obvious barriers other than the distance between vents?
Yeah, so I'm proposing that I think depth is one of the biggest barriers that we see between organisms, just because a lot of things tend to change really quickly with depth, including temperature, pressure, sunlight, you know, carbon availability, and yeah, so depth, the geographic distance, and then, you know, of course, you know, species interactions with each other. So like competition is also something that I'm kind of investigating to see if there aren't environmental barriers, what else could be keeping these organisms apart.
Wonderful. Thank you very much for the presentation. So with that, we conclude the 3-minute thesis presentations. And now at this point, I would ask our panel of judges to make their assessments and their rankings. I believe it may have just shown up, but in the chat, our participants, you will see a link to a Google form where you can go and vote for your choice for the sort of People's Choice Award, the Viewer's Choice Award, to determine who the viewers pick as their best. And so we will take a break for a few minutes while everyone fills out their forms, and we'll be back with you shortly. Okay. I am happy to announce that our judges have made their selections, man. Thank you. All right, so at last, let us have the resolution of this exciting competition. Okay, first, the People's Choice Award for Best 3-Minute Thesis Presentation goes to Jay Patil. Congratulations, Jay. Very nice job for his presentation, Magnetism from Non-Magnetic Materials Interface for Future Generation Electronic Devices.
Thank you so much.
All right. Now we'll move on to our judges panel decisions. In second place, we have Louise Borthwick with What Lies Beneath Using Geophysics on the Thwaites Glacier. Congratulations, Louise. Fine job, fine job.
Thank you.
All right, the moment you've all been waiting for. In first place, Melissa Betters, investigating deep sea snail species at chemosynthetic environments. Congratulations, Melissa. Let's get a virtual or physical round of applause for our winners and for all our competitors. It's been a fun experience for me. I do want to thank all of our presenters for participating. All the talks were really terrific, and I'm just amazed at how difficult it was for our judges to come to a decision. And I want to thank our judges. Thank you for the considerable time and passion you have put into this. Clearly, you have taken this responsibility to heart and done a great job of really carefully weighing these highly competitive talk. So thank you to our judges. Thank you also to our viewers who participated in the People's Choice Award. Thank you for taking the time to do that. Thanks also to CST Graduate Affairs for hosting this event. And I also want to thank Megan Turan-Nyquist, who did not put herself on the thank you slide, but who really coordinated all of this and did a great job putting this event together. Thank you all so much for joining us, and we hope to see you next year at this event as well. Thank you for tuning in.